Good morning, guys. Hopefully you found the previous video on mitosis quite useful. We're going to do the same again for the cell cycle lesson. So it's 6:38 on Tuesday, the 17th of November, and I'm getting this done because it took me 4 hours to upload this or the previous video yesterday. So hopefully this should be a little bit smoother because I'll pretend to know what I'm doing now, and also hopefully this video won't be as long because slide-wise it's only 26 rather than 32. So we're going to start with the starter as per usual. Have a go at these questions. Pause me now. You give yourself about 5 minutes and I'll shut myself up for a second and then afterwards we'll come back. So pause me now, have a go, and I'll see you in a sec. Alright, so let's start looking at these then. We're going to think about last lesson, our GCSE knowledge, and other things that we've learned over the course of the year, maybe the last 2 years as well. So I'm going to go back to that brown pencil. Hopefully you guys could have all seen it. If not, I'm sorry. Maybe work with a buddy. So we'll start with number 1. What's the difference between mitosis and meiosis? Now, the easiest way to do this, I find, in an exam is to do a little grid. So I know what I'm talking about. I can organise my thoughts a little bit more. So mitosis and meiosis. Now, the first thing we think about is the cells that it occurs in. So mitosis occurs in somatic or body cells, whereas meiosis occurs in our gametes. Remember, that's the word that we're using for our sex cells, sperm and the egg. Mitosis produces cells that are 46 chromosomes large, whereas meiosis produces haploid cells, which have only got 23 chromosomes. And remember, we are using the terms diploid and haploid again. I'm going to up that vocabulary that we're using. Mitosis produces 2 daughter cells. And I don't know why they're called daughter and not son cells. Probably sounds nicer. Whereas meiosis produces 4 daughter cells. And finally, mitosis produces genetically, that's the G, I'm not going to write it out, it's too early, identical cells. Whereas meiosis, through various processes that we'll look at in topic 4, things like crossing over, things like we'll just stick with crossing over for now, mainly because I can't remember. Produces genetically distinct cells. Okay, so they're slightly different from the parents. They would have swapped some genes around, and they would have become slightly unique. 4 main stages of mitosis. Just as a heads up, writing PMAT in the exam is not good enough. It needs to be prophase, and you have to be able to spell these right, metaphase, anaphase, and telophase. Notice that I haven't included interphase because that comes before mitosis, and cytokinesis, which is the splitting of the cytoplasm. Remember, mitosis is just nuclear division. Question 3, what happens during interphase, the pre-mitosis stage? We've got DNA replication, and we know how that happens because we did module 1. And then we have got things like the centromere joining our chromatids. Okay, so remember we're going to get that typical X shape now. So we've got 1 arm, 2 arms, and then we've got the centromere in the middle. We've also got growth of our cell, and we're going to have a look at that in a little bit more detail in a second. Okay, and finally, why do we need checkpoints during cell division? That's just so we can identify errors. Okay, identify if things are going well or not well. So we're identifying errors, and if not, or if the cell has got an error in, we undergo apoptosis. Reminder, that is that programmed cell death. It's a bit ruthless, but it's got to happen. Oh, what have I done? Broken my thing. Okay, perfect. So that's your starter. And let's get into the cell cycle. So today is a little bit of a weird lesson, or this one's a little bit of a weird lesson. It looks very briefly at the stages of the cell cycle, so a bit more detail into interphase. And then the rest of the lesson is looking at cancer. Okay, so as I said in the previous video, it's not... it's a very sort of brief look at cancer. It's looking at things like the risk factors and the types of tumour and stuff. Now, unfortunately, if you have got someone in your family going through cancer, it is part of the specification. You do have to know about it. But I'll try to be as blank about it as possible. I recognise it's quite an emotive issue. If you do, or if you are being affected by this personally and you don't want to listen to the audio for that part of the lesson, that's absolutely fine. Just crack open your books and try and do it from then, or the PowerPoint on Edmodo. Okay, no judgment either way. So we're gonna start with the cell cycle, as I said. Now the cell cycle is really interesting because only certain cells can divide. Okay, things like our neurons, our brain cells, generally don't divide, whereas things like our skin cells and our liver cells and our cells of like lots of other organs have to be able to divide because they're in situations that means they're going to get damaged regularly. Now, the important thing to note about the cell cycle is that it doesn't happen continuously. Okay? Sometimes the cell divides and it does that mitosis bit. Other times it's just there chilling, it's growing, it's getting ready to divide. Okay, so we'll have some periods of time where this mitosis is happening. Remember, this is an active process. It's energy intensive. And therefore we don't want it to take a long time. Okay, it's a short period of time. Again, highlighting the fact that after mitosis, we've got cytokinesis. The processes of cell growth are all in interphase. So we said that interphase is the step of the cell cycle where the genetic material is duplicated. That is true, but we can break it down even further. Okay, so just as a heads up, we've got this G0 phase here. This is the stage that a cell will go into if it can't divide. Okay, so this might be because we've got some specialised cells which don't generally divide, or it could be because the cell is damaged and we're just going to kill it. Alright, so we're going to take a look now at the 3 stages of the cell cycle. So interphase is all of the other bits, as we said, that involve the DNA replication and getting your cell ready for division. Now we say there, although the DNA is replicated, the cell is at rest. That's because it's not actively dividing. And we can split it up into these little sections. Okay, remember last time we said that mitosis, or the cell cycle, was like a pizza slice, and mitosis was a little bit up here. The rest of the pizza is made up of interphase, and parts of those are the G1 phase, the S phase, and the G2 phase. Now this occurs regardless of whether the cell is undergoing mitosis or meiosis. And this is really important because it prepares our cell and makes sure that there is everything ready to go for cell division. So we're going to start by looking at G1. Okay, now G1 is there to increase the cellular contents. Alright, so cellular contents excluding the chromosomes are duplicated. That could be our organelles. Okay, remember we've said that mitosis is an active process, so we're definitely going to need some more mitochondria. There ain't no way I can undergo cell division without lots of energy. So that's going to be producing our ATP. That's going to be undergoing respiration. And then we've got to consider what else we might really need. So if we're making a whole new cell, pause me now and have a think about what organelles you might need to make lots and lots of cell components that maybe would be packaged into vesicles. So hopefully on your discussion, your thought processes, you might have thought about ribosomes. Okay, we need to make some proteins. Alongside the ribosomes, we've got to remember now, because we've done the lesson on organelles, that we've got the endoplasmic reticulum. And I should have written ER, it's too early for this as well. Specifically, the rough endoplasmic reticulum. That's the one with all of the ribosomes on it. It looks like that. Yeah, it's got all the little bubbles on the outside. You could have also mentioned, I'm going to write this above ribosomes, about the Golgi. Okay, I'm just going to write Golgi app because I haven't got enough space. Now, all of those are going to go into our process of protein synthesis. Okay. We are then going to have our synthesis phase. Okay, so synthesis is literally where our chromosomes go from 46, if we're in mitosis, to 92 chromosomes. We're getting ready to divide. Now in G2, this is our final growth stage. Sorry, just to highlight that the G is growth. And the S is for synthesis. Okay, so we're making new stuff. So in the G2 phase, the cell is being double checked again. Okay, we love those checkpoints. They make sure that we know what's going on. We make sure that we know that our cell is ready to divide and it's not going to be damaged. So here the cell is going to double check the duplicated chromosomes and it's going to make sure that any errors have been fixed. Generally, we don't want to kill a cell in the G2 phase because we've spent all of the time getting it ready. So instead we just fix it. Normally it's just about whacking a different nucleotide in or just making a few more bonds. Okay? Now the cell cycle is going to vary between organisms, so between us and a rat, between a rat and a fish, for example, but it also varies between cell types. And that's something to be aware of because they really love maths questions relating to these. And at the end of the lesson, there is one that I'd like you to have a go at. Okay, so it varies between cell types. Now, in our cells, in mammals, it normally takes about 24 hours. So skin cells will turn over very regularly. Okay, that's generally what dust is. How disgusting. But around 90% of that time of the cell cycle is spent in interphase. So if there was a question, say, that said the cell cycle in mammals lasts 24 hours, around 90% of that time is spent during interphase. How long is interphase? You would have to do 24 times 0.9 because we've got 90%. Bung that in a calculator and it'll give you an answer. Haven't got a calculator right now because, as I said, it is currently 6:52 in the morning and I'm not about that life. Okay? But make sure we're confident in recognising the cell cycle and acknowledging the fact that it does vary. It is not a standard uniform process. It depends solely on how regularly the cell is damaged or how exposed the cell is to damage, and also how complex the cell is. Okay, so things like white blood cells don't turn over. That's what we say when they're regenerated. They don't turn over very much. Okay, because we need things like our memory cells, the ones that stay in our blood, to last for a long, long time. If we turned them over, we'd lose them and then we'd lose the immunity that we've got. Conversely, if I've got loads of old skin cells on my arms, for example, that's just going to appear as like dandruff and eczema and things like that, which is fine, but not perhaps aesthetically appealing. Now, in the exam as well, they love having mitosis as a graph like this. Okay, so pause this now. Draw the pictures for me. Okay, make sure you're using a ruler, please, or something with a straight edge. And then I'd like you to describe and explain the patterns shown on that graph. I will go through it in a second, but I would like you to have a go. Okay, so describe and explain the patterns in total cell mass and the mass of DNA per the cell varying over time. So say what you see and tell me why that happens. Pause now and play again when you're ready to look at the answers. Hello again. Hopefully, I don't know why I said that. That was really weird. Hopefully you are okay with this graph. You've had a go at the graph. Now let's see if you are ready, or you understood it. I don't know what's going on this morning. You're ready to go through it and see if you got it right. So we're going to take a look at the top graph first. We're looking at the total cell mass. So we can see that there's periods of growth shown in the blue, followed by rapid periods of dropping. Okay. Now the periods of growth of the total cell mass, so I'm just going to write this over here. So growth of total cell mass lines up or signifies interphase. Okay, now with this interphase, remember we have got the G1 phase where we're going to increase our number of organelles. That's going to make our cell heavier. No, that's horribly written. And we've also got the synthesis phase where we're going to increase the amount of DNA we've got in the cell. If we take a look at the graph below, I'm going to highlight that one in green. You can see that we've got sort of nothing going on from 0 to 12 hours. And then we've got this upshot. Okay, this upshot in mass of DNA per cell occurs or shows where the S phase is happening. Okay, so all we're doing is increasing the mass of DNA. Okay, we're duplicating our DNA then. So increasing DNA, duplication. Now hopefully you will have noticed that after that stage of DNA growth, mass of DNA per cell increasing, you will have noticed this plateau. Okay, so actually I need to rub that bit out and rub the green bit out as well, because that shouldn't be there. The green arrow for the S phase should be specifically only pointing to the growth. Okay, it should only be pointing to that synthesis of the new DNA. Now this orange section, hopefully this comes out, highlights where we've got our G2 phase. Okay, remember, we're not going to get any more growth of the cell. It's there to just double check what's happening. So our DNA remains constant. Now at this stage, if you notice in the graph above, you've still got some cell mass growth, and that might be because you're increasing things like the cytoplasm. Maybe you're taking on some more water to increase the cytoplasm, for example. Now the final thing we should have noticed is that we have got this drop, the downshot, whatever you want to call it. I'm going to highlight it in that hot pink colour that we love. Got this drop happening at the same time on both graphs. Now this drop, I'm going to write over to the the left hand side, sorry. The drop indicates mitosis, and it also indicates cytokinesis. Okay, these are occurring at the same time in both graphs. Okay, and we get that drop back down to the starting levels because our cell is dividing. So the cell mass is going to be divided between two cells now. The DNA is going to be divided between two cells now. But the key thing to recognise here is that the concentrations of both DNA and the total cell mass do not go back to zero. They simply just drop down to the point where the cells started. Okay? If you didn't understand any of that, maybe go back and have a look at the previous slides on the cell cycle. You can find this PowerPoint on Edmodo, just not with all of the notes. Or you can take or you can just rewind me and go back to those slides on the video. Maybe go back and look at the mitosis lesson as well. Okay? So this is just a little bit of basic detail. They might say in an exam question, how much of the cell cycle is taken up in the synthesis phase, and you would have to look at the growth or the replication phase where DNA is increasing. Okay, so remember this is the S phase, and this would be G2, and this would be G1. Okay, so if you wanted to, it might be worth, if you've got some highlighters, just overlaying all of those stages, rubber, not highlighter, highlighting those parts of the graph where things are happening. Okay, making sure that you recognise that these two graphs occur or are overlaying each other basically. Alright, so the final thing that we've got to look at, as I said, is all about cancer. Now, here is the point that you need to stop me if you are having family issues or you've got someone that you know or love that's got cancer. If you don't want to listen, that's absolutely fine. This lesson is on Edmodo on the big post that I put about topic 2, and it is lesson 2.9. Okay, so pause me now if you don't want to listen on, and otherwise I'm going to go straight into it. So cancer as a thing is, yes, the emotive condition that we know about and we've probably all been affected by, but it's a group of diseases. Okay, there's lots and lots of different types of cancer, and the key thing that makes cancer cancer rather than any other kind of condition is uncontrolled cell division. Okay, uncontrollable mitosis. Now, what that means is if you look at the picture at the bottom, normally if a cell divides, it just goes and makes a new cell, and then that goes and makes another couple of new cells. But sometimes the cells will become damaged, and we said that normally if the cell reaches a checkpoint, maybe the metaphase checkpoint, and it's deemed to not be okay, the cell will just undergo apoptosis and it dies. Now, what we've actually got in cancer is a stress or a stressor from the environment, something that increases the likelihood of mutations, damaging the genes. Okay, so we're going to get a mutation or a change in switching on or off that regulate this process of mitosis. And we're going to have a look a lot more at the genetic basis in topic 8. I think it's 8 next year. Okay, so in cancer, this cell that was originally damaged doesn't die, and instead what happens is it divides. So it divides, and because it's got a mutation already, it means that it's more likely to pick up a mutation. Okay, and that happens a few times, and for some reason the body hasn't recognised that it's damaged. The body's still just like, yeah, we'll keep going mitosis. Yeah, that's cool. And eventually it gets to the stage where we've got uncontrollable cell growth. And this is where we get a tumour. This is what a tumour is. That hard ball of cells is just a massive clump of uncontrollable cells that have divided. Now I want you to pause now and have a think about any environmental, not genetic because we're not looking at that today, factors which can increase the chance of cancer. And maybe if you know some specific ones, you want to have any specific factors, link those to those specific cancers. So pause now and we'll go through the answers on the next slide, but I want you to have a think for now. So cancer is mainly caused by lifestyle factors. So you can have a look at this first row, or the top row. All of those are things that people have a choice of. Okay, you choose to smoke, you choose to drink, you choose, or maybe not, depending on the financial situation your family's in, a lack of fruit or vegetables. Maybe you choose to overly indulge in things like bacon and sausages, which have been shown to have an increased risk with some colorectals or bowel cancers. A lack of fibre isn't going to help your gut. And finally, being overweight, although not directly linked to cancer, the factors that make people overweight generally go hand in hand with causing cancer. Okay, so maybe people drink too much, which leads them to being slightly overweight. Maybe they don't exercise, which leads them to being overweight. Okay, there are also other factors which we can't really control that are going to potentially cause some cancers as well. So females being postmenopausal, you see an increase in things like breast cancers because the genes that regulate the hormones are suddenly gone a little bit awry. The menopause means that we're going to stop having things like periods and stuff, and those mutations then result in ovarian and breast cancers. Ionizing radiation is generally an employment risk. So if you've ever had an X-ray or a scan at the hospital, you will have noticed that the radiographers, the nurses, the doctors who are viewing your imaging all wear those big lead vests. That's because exposure to radiation is damaging. You know from GCSE they're high-powered particles. Okay? They're high-powered particles or waves that can cause damage to cells. This is also a specific problem with people who spend a lot of time closer to the edge of the atmosphere. So pilots are exposed a lot more to ionizing radiation from the sun, for example. Occupational hazards might have an impact if you work with chemicals, fertilizers, things like that. You could be more at risk. Reproduction is a bit of a tricky one because there's lots of hormonal fluctuations that happen when females are growing, developing, producing a baby, and they can sometimes cause cancers. And finally, the standard one that we all know from GCSE is sun exposure and sunbeds. So making sure that you're wearing sun cream, reducing the chance of that UV radiation hitting your skin cells and damaging them. At the bottom, we've got a couple of examples of cancer, esophageal, colorectal and breast cancer, and the risk factors that cause them. Okay, now notice that for esophageal cancer, smoking accounts for almost 70% of all cases. Now notice that these percentages don't add up to 100, so normally people are going to be affected by a multitude of factors. Okay, it's very rare that one of these happens in isolation. Now, cancer as a process is particularly damaging because those cells are able to grow rapidly, and because of that, they are uncontrollable and can start invading other tissues, and that's where we start to see systemic damage. Okay, now the process of cancer development normally occurs in cells that have a high turnover. Okay, you see the cancers that are rarer occurring more rarely because the cell cycle happens less frequently. So the introduction of mutations isn't going to happen as often. Okay, so cancers are most commonly found in the lungs because, our alveoli need to be replaced all the time because they're going to get damaged maybe by particulates and things like that. The prostate involved in semen production in men, and the ovaries in women, causing things like gamete production and the release of an egg during menstruation. The stomach, the pancreas and the large intestine, so all of those digestive organs are going to be really knackered regularly. Yeah, your stomach's working all the time. Your pancreas is constantly monitoring your blood levels. Your large intestine with its brush border is going to be damaged over time. Now, because of that, they're replaced over time very regularly, and so the cells have a higher chance of getting those mutations that cause the cancer. Now, hopefully, you will have known that there's two different types of cancer, or not really cancer. There's one type of cancer and another type of tumour. A tumour is not dangerous, and we also say that it's not cancerous because it can't invade other cells when it's benign. Okay, benign, spelt like that. It becomes dangerous or cancerous when we call the tumour malignant. And on the next few slides, we're going to take a look at what those terms mean. Okay, so malignant tumours are the bad ones. Okay, we can look at this at the bottom. These tumours grow rapidly. They are very fast dividing. They're generally very mutated. And you'll notice here as well that they are randomly shaped. Okay, so they're poorly formed cells. They're a bit like if you bred a weird dog that has lots of different bits from a big dog and a little dog. They're just bad. Okay, and we say that they're less compact. So on an imaging, so maybe if we're using an electron microscope, for example, the tumour edges are fluffy. They're not defined. So they might just look a bit like a blob with little ripples around the edges rather than a benign tumour, which we'll get to in a second. Now, these tumour edges being fluffy or not being compact means that some of the cells around the edges are able to break off. And once they break off, they can enter the bloodstream, which is slightly problematic because then they can spread to other parts of the body. Okay, they can form what we call secondary tumours. And this in medical terminology is called metastasis or metastasis. Okay, you can say that the tumour has metastasised. Conversely, benign tumours are slow growing. So they might just be felt as a lump, but that develops over a short period of time. And they're more compact in formation. So they're actually a ball of these poorly dividing cells enveloped or contained by a connective tissue capsule. So these benign cells or these benign cancers are easily able to be removed. Right. Conversely, these malignant tumours, because of their fluffy edges and the fact they can break off, are quite difficult to be removed, and that's why we end up having to have medical interventions, things like chemotherapies and stuff, to go alongside them. So malignancy, or the development of a malignant tumour, occurs when growth factors are damaged. Okay, so growth factors are things controlling cell growth, things normally genes or gene products, which we will look at next year, controlling growth, are damaged. And this is because of mutations, maybe just something's happened over the DNA replication process and they've become a little bit broken, or stressors. So UV radiation has hit your skin cells, that's induced a mutation, then that cell has become cancerous. Okay, now normally, as we know, these mutant cells are destroyed, they undergo apoptosis, but if not, they're going to quickly replicate. They form clones of themselves, or they just replicate really rapidly, and that is the formation of the tumours. Now, for the last little bit of the lesson, you guys are going to do some thinking about cancer treatment, and then you're going to have a look at some exam questions. So, chemotherapy is the chemical, so chemo, treatment of cancer. And this is normally done by a long-term, over a few hours, intravenous drip, and you have to sit there and wait while the chemicals go around your body. Okay. Now, this chemotherapy halts parts of the cell cycle. So I want you to have a think about what parts of the cell cycle could be affected. Hopefully you had a think and you came up with some good ideas. Now, the major parts of the cell cycle that we're going to look at being affected are the following, and hopefully these line up with what we did last lesson using your previous knowledge and this lesson now. The chemotherapies are normally either spindle formation inhibitors and DNA replication inhibitors. Okay, so what I've got on this slide is I've told you the type of drug they are, and I've given you a brand name, so Taxol and cyclophosphamide. I would like you to spend some time in your own notes, I'm not going to go through it, finding out and thinking about how each of those classes of drugs can help arrest or stop cancer cell growth. So spend a few minutes, maybe 5 or so, doing some research and figuring that out. And I want you to be able to tell me on Friday how those work. Now, one of the most, one of the most difficult to accept side effects of chemotherapy is hair loss. Okay, so we're going to have a look now at why that happens. Now, cancer cells, because they're dividing so quickly, take up lots of nutrients. Okay, we say that they're really metabolically active. This is why one of the symptoms of cancer is weight loss, because these cancer cells are massively, rapidly dividing, and so they're taking a lot of the cell's energy, or they're taking a lot of the body's energy up just to keep growing that cancer cell. Now, chemotherapies travel around the bloodstream. As we said, you're sitting there with a drip in for a few hours, and they target those rapidly dividing cells. So it recognises that cancer cells are really, really active, they're dividing really quickly, and they're like, nah, get rid. And this semi-targeted intervention is why they're so effective at treating cancer cells, but also partly the reason why they cause hair loss. So spend a little bit of time thinking about why chemotherapies targeting those rapidly dividing cells causes hair loss. Pause, and when you're ready to move on, let's go on to the next slide. Now, hair-producing cells are also rapidly dividing. You'll know if you brush your hair in the morning, hair comes out on that hairbrush daily. Okay, hair on your head and eyebrows divides at a really, really rapid rate, much faster than other bodily cells, including things like arm hair, which is just slightly bizarre. Now, the chemotherapies are just going, where's the dividing cells? Where's the dividing cells? Where's the dividing cells? And accidentally target the hair cells on your head and your eyebrows and your eyelashes, thinking they are a risk. Okay, it's just an unfortunate side effect of the cancer treatment. Yeah, it's not nice. It's generally one of the things that patients find hardest to accept, but it is sort of a necessary evil at the time if that chemotherapy is going to be treating the cancer. Now, there is a new branch of medicine that's being looked at as a potential cancer treatment, and this is something called immunotherapy. Now, this is the process of using your body's natural defences, which we're going to take a look at again later on in this unit, the immune system, to fight diseases. So could you have a go at spending some time typing that link at the bottom in green into your phones, iPads? If the link doesn't work, you're just going to have to Google and find out how immunotherapy has the potential to treat cancer And I just want you to summarise one research article. So maybe find out a specific type of drug that is using immunotherapy and see what results it's got, what kind of treatment outlooks it's got as well, and just spend maybe 10 minutes widening your knowledge of chemotherapies. So pause now, spend that time, and then come back when you are done. Another bit of independent work is having a look at the How Science Works box. Sorry, that was me looking at my own book. The How Science Works box on page 81. So it's a little purple box. It's got a plus in it. It's a math skills box. I don't really understand why it starts at question 5, but there we go. And have a go at that question. The answers are in the back of the book. So this is lesson 3.8 on page... Hold on. 3.8. You're looking... Oh, I've got a misprint. I haven't got all the questions. That's a shame. You might have all the questions. Like my one starts at 5. On page 611. So answers on page 611. If you have got the misprint and you don't have the questions 1 to 4, like I don't, am I being an idiot? No, it's definitely not there. Then perhaps just skip this bit. Okay? Now the final bit that I need to tell you before we move on to the summary, the plenary, the exam questions, is your homework. So could you please have a go at all chapter 3 practice questions on pages 82 and 83 in the blue book by yourself. Okay, they're past exam questions. I want you to bring together everything that we have done over the last few weeks. It's all to do with eukaryotic cells and the cell cycle and prokaryotic cells and microscopes and all that good stuff. Could you please do it on a loose sheet of paper? Please do not tell me it's in my notes. Put it on a loose sheet of paper. Don't be an idiot. Put it on a loose sheet of paper. Don't make me ruin your notebooks. I'm not going to spend time photocopying it. If you don't have it on a loose sheet of paper, it's not handed in. Okay? Now, this is our first question. The letters A, B, C, D and E represent stages in mitosis. A is anaphase, B is interphase, C is metaphase, D is prophase, E is telophase. Write one of the letters A to E in the box to complete the statement. Chromosomes line up on the equator of the mitotic spindle in... Pause me now, have a look at the answers, or have a look at the question. And the answer is C, metaphase. Okay, so remember they're going to line up on the spindle, and then we've got that metaphase checkpoint to check that they are all of the chromosomes and the chromatid arms are connected to those spindle fibres. Next question. A student investigated mitosis from an onion tip. The student prepared a temporary mount of the onion tissue on a glass slide. She covered the tissue with a cover slip, and then she was given the following instructions. Push down hard on the cover slip, but do not push the cover slip sideways. Explain why she was given this instruction. So it's 2 marks. Tell me why she was told to push down hard, but not push sideways. So pause me now. You've got 2 minutes. Okay. Answers-wise, she needs to push hard to squash the tissue. So that's your first mark. But she needs to not push sideways. I'm just going to put not sideways. In order to avoid breaking the cells. Okay, avoid rolling the cells together or breaking the chromosomes. Okay, we need to make sure that we're mentioning the chromosomes in this answer because it said that she's specifically investigating mitosis. Okay, so we're pushing hard to squash the tissue or making it thin, but we're not pushing sideways, so we're not breaking the chromosomes. Penultimate question. Figure 1 shows the cell the student saw in the onion tissue. The The student concluded that the cell in figure 1 was in the anaphase stage of mitosis. Was she correct? Give reasons for your answer. So pause me now. Was she correct? Yes or no? And why? So you should have said yes. If you said no, that is very, very wrong. Go back to the mitosis lesson 2.7, and have a look again. Okay? Yes, it is in anaphase because the chromosomes are moving towards the poles. Okay, you could say the spindles are as well. Okay, the chromosomes are moving towards the poles. They're in two little bundles. They're in two little groups. So we can say as groups as well. And we know it's in anaphase and not telophase because the V shape, remember we said they look like little birds, shows that the chromatids are being pulled apart from the centromere. Now it's not telophase, by centromere. It's not telophase because the chromosomes are not all bundled up together. We said that we can recognise telophase from anaphase because they're all collected at the poles, but also because those chromatids, or the chromosomes now as we call them, are still really dense. They're still really visible. They haven't unwound. So each of those is one mark. If you didn't get one of them, double check, go back, maybe add some notes to yours, and carry on to the final question of the lesson, which is... The student counted the number of cells she observed in each stage of mitosis. Of the 200 cells she counted, only 6 were in anaphase. One cell cycle of an onion root tissue takes 16 hours. Calculate how many minutes these cells spend in anaphase. Show your working. So pause me now. Use the information that I've underlined and see if you can figure it out. Alright, last question. Hopefully this wasn't just a staring into the void and hoping for the answer to come and hit you in the head. We need to figure out what percentage of cells are in anaphase and therefore how many minutes that takes. So the first thing I'm going to do is I'm going to figure out how many minutes in 16 hours. Okay, and that's going to be a standard 16 times 60 because we know that there are 60 hours in a minute, 60 minutes in an hour, not hours in a minute. I'm trying to find my calculator at the same time. So if I bung that in, 16 times 60, we have got 960 minutes. And of our cells, 200 of them in total, and 6 out of 200 of those are in anaphase. Now, broadly speaking, we can then suggest that whatever percentage 6 out of 200 is equivalent to 3 out of 100. So 3% of the 960 minutes will be the amount of time it takes for anaphase to happen. Okay, so if we stick that in a calculator, you can do 3 divided by 100 times 160, or 6 over 200 times 960. I'm just going to type in 0.03 times 960, and that gives me a value of 28.8 minutes. Now it does specify in the mark scheme that you can have a mark, and I'll put that down here in this box, if anywhere you've written 6 over 200 times 960, and because you've typed it in on a calculator wrong, maybe it's buggered it up a little bit. Okay, so that's 1 mark, but you get 2 marks for 28.8, or you can have 2 marks for saying 29 minutes. It didn't specify how many decimal places to have. Okay, so this is what the calculator bungs out, 28.8, but if you've rounded it to 29 as well, that is also fine. So guys, I hope you found this useful. I hope you found going through it useful as well. I'll show you the last slide just in case. Okay, so you can stop me now, spend some time looking over your notes. If you've got to this point and you've finished, then congratulations, well done you, and spend some time now revising for me. Hey, have a lovely day, have a lovely evening, and I will see you on Friday, providing that I'm not dead. Hey, bye! If you're missing anything, it's just the answers, and we've been through all of those anyway.